Okay, well, we are going to finish lecture 4. So yes, lecture 4. So just to really quickly remind you, we left off with a secondary structure. We finished that. So we talked about alpha helices and beta sheets. There are no notes on this slide here because I had to type them on PowerPoint. So do make sure that you go back and look at the video for Tuesday. Okay, so we know that proteins, or I mentioned that proteins have 4 levels of structure. And primary, so just some recaps, the primary structure is just the amino acid chain. Okay, so that's those peptide bonds in between each amino acids via condensation reactions between their functional groups. And then your secondary structure is hydrogen bonding between the backbone, specifically between the amine and the carbonyl of the backbone, and they form either alpha helices or beta pleated sheets. So the alpha helices is going to be that twisty, stretchy structure, and the beta sheets is going to be like folding a piece of paper back and forth. And the beta sheets are going to be more rigid, more sturdy because they have a higher potential for more hydrogen bonding. Okay, so let's put BB in parentheses so we remember that it's between atoms of the backbone. And then tertiary structure, you are going to have a lot of mostly, so we'll say mostly, non-covalent interactions. So non-covalent interactions is they're not actually sharing an electron shell, right? So they're just interactions. The only exception are the cysteine-cysteine bonds. Tertiary structure are interactions between the R groups. So we can say R group interactions. So R group interacts with R group. And again, every amino acid has an R group. That R group is also known as its side chain. This is how amino acids differ from one another. So if you have an amino acid with a polar side chain, okay, that is typically going to interact. Remember, polar means partial positive and partial negative. That is going to interact with the amino acid or another molecule that is also polar. Okay, because of this partial positive and partial negative. And so this is typically going to happen mostly because the polar amino acids, if you go back to the polar category, where are we? Let's keep going. The polar category, all of them have, well, let's see, all of these ones, the ones with the hydroxyl group, have the ability to hydrogen bond. And then this carbonyl right here, this has these lone pairs. So this can also form some kind of polar interaction, some kind of van der Waals force, or maybe a hydrogen bond with something else. So the most typical type of interaction between polar molecules is going to be a hydrogen bond, but it's not exclusive. So in parentheses, we're just going to put van der Waals because that could also happen, right? But this is going to be most likely. All right, so the acidic amino acids, if we remember from that chart, I'm not going to go back, but the acidic amino acids have a formal negative charge. We know that opposites attract. We know something that is negatively charged is going to want to interact with something that is formally positively charged. And the positively charged amino acids are called your basic amino acids. Now, because they're acid-base, you can call it an acid-base reaction, but really what's happening is you have a negatively charged anion interacting with a positively charged cation. This is actually, by definition, an ionic interaction. Other people call this electrostatic. And just again, to put it into perspective, this is just talking about how the amino acid chain folds and interacts with itself. So we're talking about intermolecular interactions. But proteins can also interact with other molecules floating around in the cell. So if you have a protein with a negatively charged side chain, that amino acid can interact with other molecules that are positively charged. So just keep that in mind, that it's right now this slide is talking about how amino acids interact, but this can also be used to describe how amino acids of a protein can interact with other molecules. So polar other molecules too. Basic other molecules. Okay. And then finally, amino acids with non-polar side chains, a couple things are important about them. When the protein is all folded, they're rarely, if ever, going to be on the surface. Why? Because proteins are inside cytosolic, water-based environment. Non-polar molecules don't like that. So they are going to want to kind of sequester themselves or hide themselves, and they'll be in the center of the folded protein. So these you can find in the center of the folded protein. And since all of the nonpolar amino acids are going to be there, we like to say that the nonpolar amino acids interact with other nonpolar amino acids, kind of by force. They don't really have a choice because each of them are just trying to sequester from the cytosolic water-based environment. So they're kind of each in this little tiny little huddle, okay? And we call these hydrophobic forces. All right. Okay, so again, just to fill out this slide here, the only covalent interactions that exist are going to be between the sulfhydryl groups of two cysteine molecules. Remember, each of these get oxidized and form a covalent bond. Okay, so cysteine, you're always most likely going to see cysteine interacting via a covalent disulfide bridge. Okay, and so what drives this folding ability? I like to make up words, I guess. Okay, one of the things that drives it is the molecular properties. Obviously, if something's polar, it's not going to interact with something nonpolar. So definitely the molecular property, okay? But remember what happened before. It's kind of all happening at the same time, but remember the lower level of structure. So the first level of folding is that secondary beta sheets and alpha helices. That's already bringing different amino acids closer together to allow them to fold. So the molecular property is something that influences folding. The secondary structure brings amino acids closer so that they can fold with one another. The whole idea that certain amino acids are there matters. So not only primary structure influences tertiary structure and folding, but so does primary. If the amino acid side chain doesn't have any nonpolar amino acids, you're not going to find any hydrophobic forces. Okay. And last but not least, this is something that we'll touch on a lot through the semester, is that cells are very, very, very stingy with their energy. They only use it if absolutely necessary. So proteins tend to fold in the conformation that requires the lowest amount of energy. Okay. So, and last but not least, we also say that, you know, we're in a tiny little cell. So we can't just make the biggest giant protein that we want. We are going to make, sometimes physical space actually limits a protein's folding. So we'll say that physical space plays a role. Okay, or at least makes an impact on how a protein can fold or not. Okay. Okay, so there's your tertiary structure notes. Now let's talk about this and look at a picture. So here's just a chain of amino acids. In gray is the backbone, right? And shown here only a couple, like here is one amino acid. So there's a central carbon, and this picture is trying to indicate that the side chain is a negative side chain. Whereas this is another amino acid, there's the central carbon. This side chain is a basic side chain, okay? So let's take a peek if we are looking at this. So imagine there's a bunch of amino acids in a row, okay? This is the amino acid side chain. Okay, so let's look at that first one that we just started talking about, the amino acid with a negative side chain and amino acid with a positive side chain. And what this cartoon is showing here is just a zoom in of the actual atomic structure. Okay, so the molecular structure, the atom composition. So here's your backbone, here's your central carbon, here is the side chain that's coming off of it, and here's your acidic, this is your acidic side chain. Okay, and it is attracted to a basic side chain. So here's the side chain of the other molecule or the other amino acid. Again, the gray is just highlighting the backbone, so the alpha carbon, the amino, and the carboxyl group. And so you can see here that there is an electrostatic or an ionic interaction. And if you look at this, this in its primary structure was a big long chain, a linear chain. And so now what you've done is you've taken a amino acid on one side of the chain, an amino acid on the other side of the chain that just happened to be a negatively charged and positively charged, and you've brought it together. So you're really creating this structure. Okay. If we look at the next little interaction here, and we zoom in and see really what's going on. Okay, no. If we look at this interaction here and zoom in and look at what's going on here, here's the backbone again. And all of these, let me try to highlight these hydrophobic or non-polar side chains. There's a side chain. There's the side chain. And then here's another side chain. Right, you have all those hydrocarbons. Hydrocarbons are stable because they have similar electronegativities. They share electrons equally and the electrons are distributed equally. All of these things really can't interact with water. So you find them in the center here where your cytosol is out here, hidden in the middle. So this is what we call your hydrophobic interactions. And they're not actually making any kind of interactions here. There's no interacting. They're just kind of all forced to be squished together. Okay, now, last but not least, this tiny little, this last structure, we're gonna zoom in on that. And this is actually not an example of tertiary structure, but it's just to remind you that the backbone can also interact with each other, right? So if this is, here is, we're zooming in here and here. That's right there and here. So the backbone has been kind of closer to one another, and now it can interact via a hydrogen bond. This is not, this could be part of an alpha helix, and this picture doesn't look like a sheet, but actually it does. This maybe kind of looks like a sheet, okay? But it's just, this cartoon is really just to emphasize that within one single protein that is folded, you can have tertiary and secondary structure all happening in the same little space, or not, or a little bit of each, or not. It's just really depends on those things that we just talked about. Okay. All right. Cool. So one more kind of peek at what hydrophobic forces are. Again, here is your linear primary structure of the amino acids. The non-polar side chains are just highlighted in green just to kind of show you what that looks like, to keep an eye on that. And when this protein folds in its lowest conformation, lowest energy conformation, you're typically going to find all of the non-polar side chains in the middle, sequestered from the rest of the cytosol. And then because the polar molecules can interact with water because of their partial charges, you're going to see them sticking out on the perimeter. Right, so this little polar side chain is begging for another polar molecule to interact with, whether it's another protein, whether it's a sugar, whether it's whatever. So if this is a protein in your cell, this has the ability to interact with polar molecules. which is really great. This is what we want to happen. We want molecules to interact in our cells so that cells can undergo their processes. Okay, let me see what else is next. Okay, I'm gonna go ahead and do quaternary. We'll end with questions and then we'll switch to lecture 5. Okay, so last but not least, we have quaternary structure. Okay. Now, this is really important to remember is that not all proteins fold in quaternary structure. Okay, some do, some don't. Higher level organisms, more of their proteins do use quaternary structure. So what is quaternary structure? It is essentially 2 or more completely folded proteins that then, once they are individually folded, so if this is protein 1, it was a linear chain, it starts doing its alpha helices, and then its tertiary structure. So here's a folded protein, primary to tertiary structure, all folded in its lowest energy conformation, good to go. And then a different protein, or maybe another copy of the same protein, that part doesn't matter, we'll get into terminology. The same thing happens, primary, twisty beta sheet, secondary, tertiary. So now you have tertiary folded protein, tertiary folded protein. Alone, this does not do anything, has no function. Alone, this does not do anything. No strength, no function, no nothing. It's only when they bind together. So what did we just say in that previous slide? All the polar molecules are sticking off of the perimeter. All the polar molecules or all the polar side chains are sticking off the perimeter. Now these two things can interact. Their polar side chains can interact. And that's what brings them close together. When they're close together, then this whole thing is now ready to do its job. Okay? So if you have a protein that's all folded beautifully and perfectly but has no function, this is called a subunit. This is also called a subunit. And when the two subunits interact, that's when they form a functional protein. So this is a cartoon showing that. Here's your first protein folded in all of its structures, primary to tertiary. Primary is not folding, but it's the sequence of amino acids. Here's the second one. If it is the same, if it's two copies of the same exact protein, and once they are put together, there are 2 of them, so we call it a dimer. But because they're 2 of the same, we call them homodimers. So this is 2 of the same. Di means 2. Okay, so we'll say actually, homo means same, di means 2. So the other option is what, and this is just a dimer. So this is just showing 2 subunits. So this is a subunit, no function on its own. This is a subunit, no function on its own. It's only when the 2 subunits come together that this whole entire structure is now a functional protein. Okay, the opposite of a homodimer is a heterodimer. So one protein of an amino acid sequence folded up into its primary all the way to tertiary structure, and then you have a completely different amino acid sequence, but it is also primary, secondary, tertiary. Again, alone has no function, so we call it a subunit. And then the other one, we already know that this alone has no function. It's when those 2 subunits come together that you form a functional protein. So this is your functional protein. It's still a dimer because there's 2 of them, but hetero means different. And that's the terminology that we use for quaternary protein structure. Doesn't just mean you only need 2. So quaternary structure is 2 or more subunits interacting to make a functional protein. Okay, so if there's 3 subunits, we call it a trimer. If all 3 of those subunits are the same amino acid sequence, we call it a homotrimer. That's not very often. We don't have that very often in our cells, but we can call it, if they're different subunits, we call them a trimer. So this is a completely different amino acid sequence than this versus that. They're completely different amino acid sequence. And collagen actually is an example of a trimer. So collagen requires 2 or 3 separate amino acid sequences. Each of them need to be folded properly. Once they're all folded properly, then they come together to actually form collagen. But before that, we just call them subunits of collagen. Okay, so the primary structures are different. Okay, so this is technically a heterotrimer. And then if there's 4, tetra. So that means 4. 5 is penta, pentamer, hexamer, heptamer, and you can keep going, okay? And so sometimes you have something like hemoglobin. Hemoglobin is a protein and it is constructed of 4 different subunits, not 4 different, 2 alpha subunits and 2 beta subunits. So these 2 are the same amino acid structure. Same amino acid chain, same secondary and tertiary structures, but alone they can't function. You need just 2 copies of them, sorry. These chains are also the same, but these amino acid chains are a completely different sequence. Okay. These are just some examples for you. Right? And I'll give you one more example just to really show how this is relatable. So this is a potassium channel in S-lipidons. Okay, and so we know that certain electrolytes or certain ions have to come in and out of the cell, and this is what helps balance either the just the tonic environment, or it actually excites certain cells like neurons. So neurons, when enough ions come in, that's what initiates the action potential, and the neuron sends that message to the next neuron, and this is how we actually have behaviors and movements in our body. So the way that ions can come in is they use these protein channels that live in the membrane. And so ions just can't just slip past the membrane because they're charged, and they can't squeeze past those fatty acid tails. So they need to have like a perfect little elevator to let them in, right? And so these ions, so here is one subunit. So this is a subunit. This is a subunit. Okay. And these happen to be the same primary amino acid sequence. Okay. And so what ends up happening here is that this, it's called a protein channel or an ion channel. This ion channel can only work when you have all 4 subunits together, okay? So it's just an example that a lot of the protein channels or ion channels in our membrane really are examples of individual subunits, and they only come together when they see, oh, it's time for us to go to work. So here we are living in the phospholipid bilayer, but we know that, look at all those ions we've got to let in. All right, time to link up, time to form the actual channel, and then the ions can come in. So it's less work for the cell if the cell just says like, okay, I don't want to keep making all the subunits from scratch every single time. So can you just like hang out there, and when I need you, then please form a team and form that channel so that the ion can come in. And so that's kind of why we have this, and it's really important. It's a way for a cell to conserve energy, because to make a protein really costs the cell a lot of energy. But if the protein's already formed, the subunits already formed, less energy is required to just make them kind of come together and interact with one another. And so that's what these are 3D structures of the cartoon that I'm just showing you. So each of these colored 3D globular molecules are an individual subunit, and they're coming together to form a core. And so here's the ion, it would come right through the center. Here's a little blue ions, and then it would enter the cell. Okay, and this is shown in a different type of structure, but you can see the same thing. And this is looking top down. So if you're looking down into the tunnel or into the channel, this is what it would look like, your individual subunits. Okay. All right, so there's a really nice video that recaps amino acids. I left you the link right there. And before we get to the Scuba Club announcement, I am going to go up to see if anyone has any questions. So let's see. Okay, go ahead, Lucy.
Yeah, when you were talking about the folding of proteins earlier, you
Yeah.
mentioned that the backbone can form bonds. Does it always form bonds whenever possible? Or does it sometimes
Just
just
whatever is the lowest energy conformation. And if that protein needs the backbone to interact so that it can function and be folded properly, it will.
Okay, thank you.
Max?
On the tertiary structure slide, a
Yeah.
little further up, the hydrogen bond, it showed as a secondary structure. Why is a hydrogen bond a secondary structure, but none of the
So remember when we talked about secondary structures? So secondary structures are your alpha helices and your beta sheets. And they interact. So if we go back to that.
Because it's between the backbone only?
Yeah.
Oh, okay. Thank
you. Yeah. So yeah, I guess you answered your own question. Okay, any other Dylan?
During the quaternary structure, you mentioned polar side chains interacting. Was that just an example, or is it only the polar side chains in quaternary structure?
Oh, that was just an example. Great, quaternary, sorry. Yeah, so imagine this. So let's see, let me go over my quaternary structure. Okay, so let's say that there is this, I'll draw it here. Here's your, this is somehow has some partial positive. Here's your quaternary structure over here. This one has a partial negative. These ones definitely can interact with one another, right? That's the definition. But the whole idea of these hydrostatic interactions can also occur too, right? So you have a positively charged one, a negatively charged one. This can be an electrostatic interaction. Not only that, but what about if you had cysteine and it had a little SH and you had another cysteine with a little SH? These two things are going to interact and form a covalent bond. So in quaternary structure, you can definitely have polar interactions. That's a great question. Thank you. Ionic interactions. And you're going to have your covalent disulfide bridges. But you probably will never have hydrophobic forces because the hydrophobic amino acids are going to be in the center of that 3D structure.
Thank you.
You're welcome. Okay, and then that reminds me, see how important it is for the correct amino acid side chains to be sticking off of the perimeter. So what other things affect the molecular properties? So things, because for lack of better words right now, factors. Factors that affect binding. And that this is not just factors that affect binding in quaternary structure, but also tertiary structure. So we know the molecular stuff, but to add to that list, don't forget, pH is going to matter. So pH is going to affect the molecular properties. If the molecular properties change, it affects the ability to interact. Not only that, but certain concentrations of ions and our favorite, heat. Right? All these things matter because if you heat something too much or you change the pH too much or there's too many ions and you're changing the molecular property too much, what ends up happening is that the protein slowly but surely starts to unfold. The unfolding is called denaturing. Okay. And this unfolding, obviously, if fold in structure equals function, if you start unfolding the protein, you're starting to ruin the function of the protein. And so cells have mechanisms to recognize denaturing and stop it before it gets too bad. And these mechanisms of detecting and repairing, that is called chaperone protein. So it's a whole different class of proteins. The protein is like, oh my God, that special enzyme is starting to unfold. I better go over there and help it. Okay, chaperone proteins. But if they recognize the damage when it's too late, like if the protein has started to completely unfold, there's nothing the chaperone can do. The protein will continue to be unfolded and then it will be degraded completely by the proteasome. So denaturing is unfolding. Degrading, that means you're actually starting to cut the peptide bonds apart. Okay, denaturing is just kind of disrupting the R groups interacting, the secondary structure, and degrading, that is pretty much breaking apart the peptide bonds in the primary chain. Okay, awesome. So having said that, Annika, let me have you come and share your face and
Hello. So my name is Annika, for those of you who don't know me, and I'm the vice president of the SBCC Snorkel and Scuba Club. I'm actually going to put in the Zoom link for the meeting. That's going to be next Thursday, so 1 week from today, from 5 to 6 p.m. And so next week we're going to have a guest speaker. He's a geology professor at the University of South Florida, as well as a National Geographic photographer and a cave diving instructor. He's super cool. I'm really excited to have a talk with him.
what's his name?
His name is Jason. Jason Gulley is his name, Gulley. And he's just going to talk about kind of what he does. And he does a lot of research in moulins, which is like big ice formations, and as well as like geological cave mapping. Really cool. So if anyone's interested, please feel free to message me privately and we can chat after lecture. And you can email me at sbccscuba@gmail.com. And please invite all of your friends. Anyone is welcome. People who aren't science majors, we really want them to come because we want to just share this cool knowledge about the world, about the ocean, about scuba diving. Yeah, so I'm excited about that on Thursday.
Okay, awesome. Thank you. Okay, so with that, I'm going to go ahead and change this over to lecture 5. Okay, and so now that we have a very good basis for what proteins are, how they are constructed, how they are folded, why it's important for folding with regard to function, we are going to dive into, one very important class of proteins called enzymes. And this is the biggest class of proteins. If you looked at all the proteins in our cells, most of them are going to be enzymes. And so it's really important for us to talk about what these enzymes do. And so a lot of you come in with this chemical or chemistry perspective of enzymes. We're going to use some of that to understand the biological or the cellular perspective of enzymes. Okay, so there's your lecture objectives. So in general, we all know what enzymes do. They catalyze chemical reactions. Okay, so now I'm going to ask you to try really, really hard to remember that we're inside of a cell. So they don't just catalyze chemical reactions. In this class, we're going to talk about them as them catalyzing the chemical reactions of cell processes. Okay, and when you say catalyze, we're basically saying speed up. Without enzymes, so I know in chemistry you talk about spontaneous and non-spontaneous reactions. I'm here to tell you that in a cell, very, very few reactions are spontaneous. All the cellular processes are composed of a bunch of chemical reactions, and all of them are going to require enzymes. Things don't typically happen spontaneously. In a cell, it's all very regulated. Regulated means the cell knows exactly when it should do certain reactions or not. Okay. And without enzymes, the cell doesn't have enough energy to actually drive that chemical reaction to completion. So without certain enzymes, the reaction doesn't happen, which means all of the downstream reactions don't happen and that process stops. The cell gets sick, it implodes, and that's that. Okay. And so in general, here is the overall workflow of an enzyme. So you have your enzyme, which is this big folded protein. Sometimes it's tertiary structure. Sometimes this is actually a couple different subunits. So let's pretend this is a couple different subunits. These subunits have to come together. Most enzymes, at least in humans, do exhibit quaternary structure. Not all of them, but they do. So they come together, they fold properly, perfectly to form this thing called an active site. The shape of that active site and the tiny little R groups that stick off are imperative, because what happens at that active site, that's where the enzyme's substrate will bind. So number one is that a substrate will bind the enzyme in the active site, okay? And so this substrate, this can be whatever kind of molecule. It can be a small peptide. It can be a sugar. It can be a nucleotide. It can be whatever. So all molecules also have molecular properties. So maybe there's some partial positive sticking out here, or partial or formal positive, whatever it is. There is going to be molecular properties on that substrate. And it's the attraction of those, I guess the surfaces of the substrate in the active site, the attraction is what allows the substrate to bind there. That is why folding of proteins is so important. You have to have the right R groups sticking out on that active site. Okay, so once the substrate binds, we call that the enzyme substrate complex, okay, or ES. You'll see me writing it like that sometimes. And only in this position, or conformation, I should say, we say the enzyme is active. Okay? Here, the enzyme is inactive. When no substrate is there and just the active site is inactive enzyme. The enzyme's not doing anything. It's not stealing this energy from the cell. As soon as the substrate binds to the enzyme goes to work, right? So what does the enzyme do? Its job is to modify. And what does modify mean? Well, the enzyme can modify the substrate in a couple different ways, but basically it's rearranging the molecule. It's either breaking it or like putting something on it or just shuffling around the electrons, but it's modifying it. So it's modifying the substrate. And we'll talk about certain mechanisms of modifications in this lecture if we get to it, okay? Once the enzyme is done doing what it has to do to that substrate, the result is product. So there is some, along the way, as it's modifying, there's kind of like, I don't want to call it a halfway point, but somewhere along the way, that molecule that's being modified can no longer be considered the substrate. It has changed too much. So now we call it this transition state. So you modify, the enzyme modifies the substrate to this thing called a transition state. It's kind of like a midway point, okay? So no longer looks like the substrate, not quite yet the product the cell needs, transition state. And so I abbreviate S star or S asterisk. Okay. The S asterisk, or the transition state molecule, still gets modified by the enzyme because the enzyme's work is not done yet. Then it finally becomes the product P. Okay. Sometimes the product is two products. Maybe the job of the enzyme was to break the substrate in half. Okay, that's fine. That's an option. Then once the product is made, the product doesn't have an affinity for the active site anymore. So here, different color. The reason why it interacts with the substrate is that the substrate and the enzyme, active site specifically, have a high affinity. Affinity just means attraction. The molecular surfaces are attracted. They have a high affinity for one another. That's how they come close together, and that's how they bind. Once you start shuffling things around, you're changing the molecule's properties. So you're actually changing the affinity, and so you're actually lowering the affinity. So the products here have a low affinity for the active site. And because it has a low affinity, products are like, peace, I'm out. You're not attractive to me anymore. I'm going to go away. Okay. So But the enzyme's like, bah, that's fine, no worries, because my active site is open and I can go hang out with another substrate again if I need to. If the cell needs me to do that, I'm here for that. Okay, so the active site is open again. So here it is, open again once the product leaves, and the process can repeat. So let's draw a big arrow. We can come back over here and repeat this process. So the cell really just has to make the enzyme once. And then as long as it doesn't denature or degrade, it's good to go. Great. Maybe the enzyme, if it was in quaternary structure, kind of separates into its subunits until it's like, oh, there's the substrate, subunits, let's all congregate. All right, let's do it. That's the overall workflow. Now we're going to get into detail for each of these steps. Okay. So here's the chemistry review that everyone should know. And this just kind of drives home the idea of energy. So what is something that's endergonic versus exergonic? And that's going to be different than your chemistry terms of exothermic and endothermic. Okay. So at any given point in a cell, there is a level of energy that is available. Okay. So here's what we have. Here's your substrate. The cell needs a certain product. Okay. So maybe we're going to even highlight this a different color. The cell needs a yellowish ball, not a green ball. Okay, and so the cell's like, yo, enzymes, I need you to make that green ball. And the enzyme's like, I can't. If that was the case, the substrate, in order for it to reach its transition state, you're changing it. So that requires energy, energy for everything. Right? So you're actually using some of the energy so that this substrate can reach this thing called the transition state. So this right here is the activation energy, the energy needed to get S to this transition state. Okay, so this should actually be, the activation energy is S to the transition state. Once the molecule is in the transition state, the chemical reaction will complete. Okay. So once you get S, maybe now it's like half greeny yellow, okay? Once it's there, the cell can complete the entire process. And so then you have a drop in energy from here all the way down to here in this reaction. Okay, and so the difference is this chunk right here. You've actually given off energy. So this is all the energy you needed. Okay, so that's energy needed. And then the drop is all the energy released. And so you can see by just looking at the length of these arrows, more energy was released than was needed, okay? And that it's this amount in the blue circle. That's how much extra energy was released. That is the net release energy. So because you released more energy than you needed, we say that this is exergonic. It is a net exergonic reaction. You gave off more energy than you needed. Okay, but what I'm again going to remind you is that most reactions in a cell, in a organismal cell, they cannot get to here. It requires too much energy. Most cellular chemical reactions cannot reach their transition state. So if you never get there, you're never going to complete the process. That's why enzymes are important, because what do enzymes do? They help the substrate get to the transition state using less energy. Okay, so again, there should be less energy needed. So you can see the amount of energy needed is drawn by an arrow going up. That's the amount of energy needed by use of the enzyme, because it's catalyzing that. The enzyme is actually like, I'll do it. I'll rearrange this for you. In the left hand over here, you want the S to spontaneously turn into a product. That's not, it's like, what? No, that's crazy, okay? So the enzyme is like, we're not just waiting on chance and spontaneity. I'm just gonna do it. We're busy. So I'm gonna change it. I'm changing, modifying, transit. Oh, I'm here, transition state. And then you make the product. Cool. So you lower the activation energy. And so that is a big piece here. The enzyme lowers the activation energy. And because you do that, let's see. You still make the same exact product. You're not making a different product, but you are actually going to, you're not changing the exergonicness of this reaction. You still are releasing more energy than you put in, okay? So both of these are exergonic reactions, but the only difference is that the enzyme lowered the activation energy. So activation energy is sometimes written as capital E subscript lowercase a, right? But what we call this exergonic is a net release of free energy, and free energy is delta G. Okay, so the delta G doesn't change. This is delta G, negative delta G. This is negative delta G because more energy is released than needed, more energy released than needed. The enzyme just simply lowers the activation energy. Okay, the product is still the same. The shape of the transition molecule is still the same. So it just happens quicker. Okay. So this is kind of putting those two graphs together in one simple cartoon. So again, enzymes speed up chemical reactions, and this one's slightly different than the one before because here, reactants slash substrate. Okay. Here you have your substrate molecule at some point. Here's the transition state, and here's your product. Okay. This time, the amount of energy needed, so let's just draw it, the amount of energy needed was this big chunk of energy. So this is your activation energy. And then the amount of energy released was less. Okay, so here's the energy released. If you need more energy than you release, the reaction is called endergonic. If you release more energy than you need, the reaction is called exergonic. So in this case, you needed more energy than the energy released. This reaction is endergonic. The enzyme will never change something that's endergonic to exergonic. So if we look here, without the enzyme is the solid line. Okay, so a lot of energy was needed. This would never happen in the cell anyways, too much energy needed. Well, let's say it did somehow. Okay, it's an endergonic reaction. Enzyme comes in, it's like, all right, don't worry, I'm gonna change and modify the substrate for you. We're gonna get to that transition state molecule quicker, which means we'll use less energy, and I'm still gonna make the same product. But you can even see here that if we use, let's see, let's do pink. If you had the enzyme, so pink is gonna be the enzyme, you still need more energy than you're releasing. Okay. So enzymes do not change exer to endergonic. That doesn't happen. So this is an example of endergonic. Okay. Okay, so what do these reaction rates depend on? So reaction rate is how fast, because we keep saying, oh, enzymes speed up the reaction. Okay, great. So is that the only thing that reaction rate depends on, is just if the enzyme's there or not? Well, no. So it is important to write that down. So the presence of enzymes is one thing that does affect the rate of reaction. Okay, but concentration of things also matters. So let's take a peek at what concentration of what would affect the reaction rate. So here on this graph, let me just explain what we're looking at. Here's your substrate concentration. Okay, so 0, so you have no substrates. And then here you have 1,000,000 substrates. So here we'll just say lots. And the substrate is the molecule that needs to be modified to become the product. Okay. So on the y-axis, what do we have? The initial rate of reaction. So just the speed at which this reaction completes. So obviously, if there's no substrates, there's no reaction, so the rate is 0. Okay. And it's kind of paired with, or I paired it with this little cartoon at the bottom. So the substrates are going to be the little blue diamonds. So let's draw that. Okay, your key. Your little blue diamonds are the substrate. And then your enzymes are the red shade. Terrible. Okay. So if you have a low amount of substrate, the rate is low. And if it's a healthy cell, you have way more enzymes necessary. So in this little case right here, we'll just say we have a lot of available enzymes. Maybe they're not all used, but they're there in case we need them. Okay? And as the substrate concentration increases, you still have those available enzymes. So we still have some fewer. So we have lots of available enzyme, fewer available enzyme, but still there. So we can still hurry this reaction up, okay? And so that's what we're seeing is that if you have these enzymes there and as the substrate is present, the enzyme is modifying every single substrate it's binding to and releasing it, and then it's open again and it takes another one and it releases it. Okay, but there's some threshold, and that's this threshold right here, where at this point, there's no more available, not no more enzymes, just no more available, we'll call them active sites. Okay? And you can see that in this little picture here. All of the enzymes present are busy. They're busy doing work. And now there's more substrate than active site. So at this point, there's more substrate than active sites. And there's no possible way to keep making this reaction go faster. This is the maximum that it will ever go. Okay? And that's why this says right here, all enzymes present are working at a maximum rate. They're busy. Okay? Their active sites are busy. They're actively transitioning that substrate to a product. They're busy. Okay? So not only does reaction rate depend on the presence of the enzyme, it depends on the concentration of the substrate. So you increase the substrate, you initially increase the rate of reaction only if available active sites. Which means that the third thing that is important for the rate of reaction is available active sites. Okay. Great. So I think that's a good time because I think we're going to start, yep. Questions on what I presented? Awesome. Okay, so we've talked about kind of the kinetics. That's what the graphs are and the rate and all this stuff. So we talked about the kinetics of enzymes and how they actually get a substrate to the product and the energy that it takes, yada, yada, yada. And now we're going to talk about how do the substrates actually bind into the active site. Okay, so we're talking about molecular interactions between enzyme, here's your enzyme, and substrate. How do these two things interact? And so there are actually two models for this. The first model that I'm going to present is the original model that we thought was happening. And remember, science changes over the years based on new information that we acquire. So that's why I call it the old model or the OG model, the original model. But, you know, as science progressed throughout the years, we learned that it's actually not, sometimes the old model exists for some enzymes, but most enzymes use this thing called the induced fit. So the old model is like a lock and key, and everyone knows what a lock is. Everyone knows what a key is. If you still drive an old car like me, you actually still have to put a key into the ignition and turn it, okay? But everyone lives in a house and has a key with a door, you have to turn the key. Okay, you can't just use any old key, you have to use the key. All right, so that's the same idea here where the enzyme is the lock and the substrate is the key. Because substrates in general are much smaller than big, giant, quaternary or tertiary structured proteins. So that's something to keep in mind, that the size of the substrate is always much, much smaller than the size of the enzyme, right? Because it has to fit in just one tiny little pocket of the enzyme. It's not a complete match like this. It's like one tiny little pocket. So this is, the substrate is the key, the enzyme is the lock. And so let me just zoom in here. And so hopefully what you can appreciate here is that the shape of the key is the exact shape of the active site, the surface of the key or the substrate, right? So you have that perfect exact match, which indicates, if you could even zoom in more, remember it's two molecules interacting. So they have a lot of surface area to interact. All these little black dots are molecular interactions. That is a lot of interactions. You're like, yeah, perfect match. I love it. Okay, but remember what the job of the enzyme is. The job of the enzyme is to temporarily interact with the substrate, change it all around, and then release it. Right here, this affinity is like 100% awesome. This is a super high affinity. Great. But the enzyme has to release it. If it changes the substrate to make the product, it has to change it so much and lower the affinity so much that it can actually release the product. Otherwise, the product's like, I'm cozy. Thanks for changing me around. I like it here. I'm gonna go ahead and stay. So that kind of defeats the purpose of the enzyme. Enzyme's like, no, go. I fixed you, so leave. Well, if the affinity is favorable, it's not gonna happen. Okay, so what ends up happening is that it takes a lot more energy from the cell. So the enzyme's like, oh God, here I am trying to lower the activation energy, but this affinity is so high, I'm trying to like release it. Oh gosh, I'm really using a lot of cell energy, but the cell really needs this product. So, okay, I'm just gonna have to use some of the cell energy, and that's that. So this model really is not for all enzymes. This model is when you have one specific enzyme with a very specific name, and this one enzyme only binds one substrate. Okay. So the example here is the enzyme that binds sucrose. Okay, so this is sucrose. It binds to the enzyme, and then the enzyme's job, so the cell needs a molecule of glucose and a molecule of fructose. Here are the products. This is what the cell needs. It doesn't have these, so it says, anyone out there, can you help me make them? The enzyme's like, yeah, give me that sucrose, I'll do it. Here's the substrate. Okay. So in this case, only one enzyme binds sucrose so that it can cut the bond between glucose and fructose. So sucrose actually is a disaccharide. So it's two sugars. Okay, so I'm just gonna roughly draw this. Okay, here's your glucose, here's your fructose. They are linked by a covalent bond. This whole thing is called sucrose. So the enzyme binds the sucrose, and then the enzyme cleaves that bond. That's the job of the enzyme. Okay. But we started learning that enzymes have the ability to bind a couple substrates. The substrates all kind of look the same. Like you'll have an enzyme that binds maybe disaccharides in general. That's a whole different enzyme. So it's not just one substrate. It has the ability to bind a few substrates. And that is called the induced fit. So the induced fit model, the active site, let's highlight this. Here's the active site. You can clearly see that the active site is a different shape than the substrate. Okay. But the affinity is high enough or favorable enough that the substrate can in fact bind to the active site. Okay? Not the enzyme in general, the the enzyme's active site. And so the favorable affinity, so you can see a couple of little points of interaction. Maybe this is a weaker interaction. Molecular interactions are still occurring, but much fewer of them and much weaker. Okay, so here what we have are fewer interactions, weaker interactions, but not so weak that the enzyme can't do its job. Now the substrate can come in, bind, because remember the whole job is to bind temporarily, get modified, get prettied up, and then go out. So the enzyme has the ability to modify the substrate. So here it is modifying the substrate. You see it's already like changed its color. Great, okay? So now what ends up happening is once the product is made, so the product is made, and in doing so, you've lowered the affinity. You lower the affinity by breaking interactions. And now the affinity is so low that the product gets released. And the active site is ready for another substrate if that's what the cell needs. Okay. And so one example of an induced fit molecule is a lysozyme. So the lysozyme would be the enzyme. So we'll say lysozyme. Okay. It binds polysaccharides, not one specific polysaccharides, but a group of polysaccharides. Okay. And it can bind the polysaccharides of various bacteria. And so this is, a way for us to get rid of bacterial pathogens. We're like, well, that's a weird sugar. We don't make that. Okay, let's degrade that. All right, cool. So that is an example of an induced fit. Okay. So I just want to kind of zoom in on what's happening between the substrate and the enzyme's active site. So there's some terminology here that is sometimes a little confusing because it's used interchangeably. So this cartoon shows you terms of general proteins, okay? So enzymes are not the only proteins that can bind things. Other proteins can do it too. So in general, you have a general protein. If that protein has a binding ability, we call the place where it can bind other molecules, we call it the binding site. And then the molecule that binds to the binding site is called the ligand. So all those things with the red arrows, those are general terms for any protein that can bind a molecule. Okay? And the molecule it binds is called a ligand. But specifically, enzymes are a type of protein. So the binding site of an enzyme, we specifically call it the active site. The binding partner of an enzyme, we specifically call that a substrate. So go back to thinking about the protein channel that was in the lipid membrane. As the ions are coming in, we do not say the ion is a substrate for that protein channel. We do not say that. We say that the ion is a ligand, okay? Because that protein channel on the membrane is not an enzyme. It's just a channel. It's a completely different category of proteins. Okay, so now that that's out of the way, let's look, and now let's pretend that this is our enzyme. This is our substrate. Okay. When that substrate binds, it's going to bind via non-covalent bonds. This should make sense because the job of an enzyme is to temporarily bind with the substrate. Non-covalent bonds are weak interactions. If you bound covalently, like let's just say there was a bunch of, I don't even know, somehow this had a bunch of cysteines and this had a bunch of cysteines, you're not breaking that thing apart. Those are covalent strong bonds. Okay, so it makes sense that most of the bonds in between substrates and enzymes, and even ligands and general proteins, they're going to be weak, non-covalent bonds. Not so weak that the substrate just like willy-nilly comes and goes, Strong enough to stay there temporarily, but not so strong that it's going to stay there forever. And so this here, I like, it's the space fill model. It just really shows you the size difference between a substrate molecule and a huge enzyme. So all the green molecules are the enzyme. And here's the substrate, it's this tiny little thing that just fits in a small little pocket. Okay, so you don't want to get buried and covalently keep it there for forever. Okay. So now we know in general that substrates bind to the active site via non-covalent, not so numerous interactions. So now when we say that the substrate is being modified into a product, so what exactly is happening to the substrate? How is it being modified? So this is going to show you mechanisms of substrate modification. Okay, there's different, and we've covered a little bit in some of the previous examples. So here is one way. Here's the substrate. Let's zoom in so we could just see that. Here's the substrate, and we've already seen this example. It is a molecule, it's a disaccharide. It binds into the active site. The active site cleaves the glycosidic bond. In order to do that, you're going from a polymer to a monomer. We're starting to put these concepts that we're learning, we're starting to put them together. So water is required. This is actually a condensation reaction. Oh, sorry, no, it's not. Hydrolysis reaction, right? Because you need water to break the bond, okay? And we're going from a polymer, which is the disaccharide, to two monomers, which is the monosaccharides, fructose and glucose. So one way that a substrate can be modified is by breaking a covalent bond and creating two products. We'll just say, sometimes it's even more. So we'll just say more than one product, okay? That's one way that you can do it. So essentially this is also called metabolizing. You're metabolizing that sucrose, okay? The kind of the opposite of metabolizing, so instead of breaking something down, you're putting something together. And so some enzymes do bind two substrates at a time. So here are two substrates. I don't want to circle it because I don't want you to think that's the second type. So here's just two substrates, number 1 and number 2. Okay. The cell needs a certain product that is those substrates put together and linked together permanently. So the enzyme's active site is really cool and can bind both of those substrates at the same time. High affinity for one another, not too high, high enough to bind so the enzyme can modify and start bringing those molecules together, right? These molecules right now, they're maybe far away in the cell and they might have an affinity for one another, but they're so far away. But they're like, whoa, I love that enzyme down there. I'm gonna go bind to it. Whoa, me too. So the enzyme brings them together. And once the enzyme brings them together, the enzyme allows those two molecules to interact to form the product. The product, obviously, way lower affinity. That thing does not fit into this active site anymore, so it gets released. So that's synthesis because you're making something bigger, okay? And last but not least, you are basically just rearranging the structure of the current substrate you have. And so this is really showing the actual chemical reactions that you'll be used to seeing more in this class. You're not going to see the shape of the enzyme every single time and then something coming and something leaving. So you should get into the habit of seeing that there's going to be a substrate on one side of the reaction, the product on the other side of the reaction, and the word that is underneath or maybe above the arrow is the enzyme. And most enzymes end in ASE. That's how you know it's an enzyme. And the name of the enzyme also tells you who it works on. So this word right here tells you that this enzyme works on a class of molecules called phosphoglycerates. And then if you look at the other part of the name, mutase, doesn't that sound like mutation a little bit? Mutation is just changing something. So it is. So this enzyme is changing a phosphoglycerate. So what phosphoglycerate in this example is it changing? It is changing 3-phosphoglycerate. Why do we call it 3? Because this is carbon number 1, this is carbon number 2, this is carbon number 3. Why do we call it phospho? Well, because there is a phospho group on there, okay? Glycerates the rest of the molecule. So it took this molecule that had a phosphate group on carbon number 3, and all it did was move it. So it moved it from carbon number 3 to carbon number 2, which is why we call the product 2-phosphoglycerate, because now the phosphate group is on carbon number 2. So we're not showing all the details here. You can clearly see that this was a hydroxyl group, and now it is no more, it has a phosphate group. So the enzyme is actually shuffling around electrons, right? It is shuffling around electrons so different parts of the molecule can interact with one another. So, we'll say reorganizes electrons. Okay, so here is the final version, and you have your product. So that's how picky the cell is. Like, okay, I don't want the phosphate on carbon 3. Can you please move it to carbon 2? I need that one. It's like, really, dude? Yeah, really. Okay, that's how specific and detailed our cells are. That's why I ask you to be so specific and detailed too, because the details matter. Okay, so here's a cartoon kind of summing up the previous slide. Here is an enzyme taking 2 individual substrates, bringing them together to make one product. Here is an enzyme that binds the substrate, and in this case, it's actually, in order to break that into 2 things, it's actually bending the molecule and it's going to snap in half. Okay, so it's actually forcing it, physical force. And then here is the last example that we just talked about, where the substrate binds and the enzyme is just rearranging those electrons just a tad. Okay? And the way that you can rearrange electrons is maybe you had something that was a neutral, just partial positive and negative. Maybe you had something neutral and you shuffled around those partial charges. Maybe you had partial charges and you shuffled them around and broke little bonds here and there, such that now the product has formal charges, or you did the opposite. Or maybe you just shuffled around a functional group to a different location. Okay. So these are the 3 ways that enzymes modify substrate into their transition state and then further into their products. Okay, let me see what's next. Okay, I'm gonna keep going and I'll stay behind and ask the questions at the end, okay? So what are some of the molecular factors that decide whether or not the substrate fits into the enzyme's active site? And we pretty much just covered this when we finished lecture 4. So again, if you look at this enzyme here, right, you have the active site with these R groups that are sticking out, right? Here's the unfolded protein, and then you have all your different levels of folding, okay, maybe quaternary structure. So we'll just put that in parentheses, but definitely secondary, tertiary, maybe quaternary. You have these folded proteins, the R groups are sticking out of the active site. So if we zoom in on that, okay, so we're just zooming in on this active site right here. And so each of these little bulbs are color coded. And so you can see that there are different types of R groups. And so this happens to be a basic side chain, right? And this happens to be a acidic side chain. And so you can already guess what could interact there. I don't even need to tell you. This is a hydroxyl group. So this is partial positive, partial negative. This is also a serine, so it's the same story here and here. Okay. This is a hydrocarbon, so you know that it's going to kind of be as best as it can. It's like, I'm good. I'm not going to be hanging out at the perimeter. I'm just going to try to hide back here. Okay. So if we go to the next slide, here is our substrate. Okay, so I'm going to highlight the substrate. Right, so here is the substrate. All in red. And this substrate, you guys have seen AMP before. This is a nucleotide. To really drive home the idea that enzymes, or these R groups, these side chains, don't have to interact with other side chains of proteins. They're just molecular properties. So they can interact with any kind of molecule. So that has an affinity, has a favorable affinity to it. So a substrate can be, again, any small molecule with a favorable affinity to the enzyme's active site. Right? And that cuts down your options, right? So in that active site, there's very specific R groups that stick out. Not any molecule can fit there. The affinity has to match or has to be favorable. And so we'll just look through a couple of them. So as you can imagine, this is your basic side chain. It's going to interact with a formally negative side of the substrate. It's not a negative side chain. CAMP is not it's not a peptide. It's not an amino acid. It's just finding a negative charge, and it's binding there. Substrates don't always have to interact with R groups. Here's a prime example. That is the amino of the backbone. This is the backbone. The backbone has the ability to hydrogen bond because this is partially positive and this is partially negative. So it did. The substrate binds not only to the R groups of the active site, but also to the backbone. Okay? So substrates can bind to amino acid R groups or the peptide's backbone. We don't have to go through each of them because I think we have covered this topic pretty in depth, right? So there's another ability to, and here's another side chain. This is interacting via hydrogen bond. Okay, so you still have all those different types: electrostatic interactions, hydrogen bonds. This is how substrates at a molecular level, if you really zoomed in, when we talk about favorable affinity, this is what we're talking about here. Okay, so the next thing is just an example. And I bring up this example because we are going to actually talk about it in more depth later. But this is how complicated it is. Not complex, complex. Okay, so here's your molecule of ATP. We know what ATP is. We know that it is adenosine triphosphate. We know that this is the energy, the molecule that cells use to break, that releases energy. We know that there's these high energy storing bonds called phosphoanhydrate bonds. We've covered this already. So this is our magical molecule. Okay. So it's not the AT molecule itself that is the cell energy. It's once we break that phosphoanhydrate, breaking that bond, just imagine all this energy released into the cell. Okay, so when you break this first bond, a bunch of energy is released. But other things have happened too. Bunch of energy is released, but you've also changed the molecule. The molecule is no longer ATP because you've broken one of those bonds. The molecule is now a DP, D for di, meaning 2, P for phosphate. So when you break one phosphoanhydride bond, what you actually did was you cut off one of the phosphate groups. So this is what we call inorganic phosphate, if you didn't have that. Okay. And you have your ADP And you have a bunch of energy released, more energy released than was needed to break this bond here. You still needed some energy to break it, but you released much more. Okay? So in order for ATP to be broken, you need a special enzyme. That special, and so ATP will bind to the enzyme. The enzyme needs water to do this, so this is also an example of a hydrolysis reaction. Okay. In doing so, you break off one of the phosphates, so you have a phosphate group. The remaining molecule is now called ADP, and then you have a bunch of energy. How much energy? 7.3 kcals per mole. So this is an exergonic reaction. And the type of enzyme, I'm running out of space here, is called an ATPase, okay? Because it's breaking ATP. It's in a type of, it's in a category, hydrolase. It's in a category of enzymes called hydrolases. Hydrolases are enzymes that use water to break apart molecules. That has categories. There's ATPases, GTPases, nucleases, proteases, and all of them cut certain types of molecules. Okay, keep this in mind. So first, in order to get energy to now do the next step, first we needed to cut and break apart an ATP. You're like, oh man, I just worked so hard making that. That's the whole point. Make it to break it so that you get energy. So here it is. Here's your ATP plus the water you needed, plus the ATPase that actually breaks the ATP. This process is called ATP hydrolysis. The reactants, or I'm sorry, the products are ADP, your inorganic phosphate that just goes into the cytosol. It's like, all right, I'll just hang out in the cytosol, no problem. ADP is like, yeah, me too. And then when you're ready to put a phosphate group back on me, I'm here, just let me know. Okay, so they go back to the cytosol floating all around. And here's your energy that you have. So cell's like, cool, what can I do with this energy that I just made? Okay, so, oh, well, I'm trying to actually make more, I'm trying to do a cell reaction. So I need that energy actually. And so here is the substrate. So glucose is the substrate. The cell's like, yo, enzymes, whoever's out there that can help me. I don't need this glucose. What I actually need is a phosphorylated glucose. Can anyone help me out? And I don't want the phosphate group any old place. I want it on carbon number 6. So here's carbon number 6. 5, 4, 3, 2, 1. So enzyme, hexokinase is like, I could do that for you. Give me the glucose. There it is. So here's your substrate. Glucose binds to the hexokinase. So that is the name of the enzyme. And again, the names really tell you a lot about the enzyme. So here is your clue that it's an enzyme. What kind of enzyme it is. The word kinase is going to be putting on a phosphate group somewhere. Okay, there's your somewhere. Okay. And hexo, it's going to do this to a hexose, which is a 6-carbon sugar. Okay. So the hexokinase is like, well, glucose, all right, 6 carbons, cool. All right, you'll work, come on in. And then it needs that phosphate group from somewhere. Things don't magically appear, but if you remember, that phosphate is like, I'll just hang out in the cytosol till someone needs me. Hexokinase is like, yeah, I need you, get over here. So it uses that phosphate group from the previous reaction so that hexokinase can actually do its job. So the job of hexokinase is to modify glucose. How? Put on a phosphate group. Then your brain should say, like, where did it get that phosphate group? Well, it needed it from, it got it from the cytosol, but where did it come from the cytosol? Well, that means some other enzyme must have cut ATP open or cut a phosphoanhydride bond from ATP. And so this reaction, the enzyme doing that, this actually is an endergonic reaction. Like, oh man, so all right, I did that for you, but we ended up using more energy than probably you wanted. But Not really, because the amount of energy that was released from the previous step provided the energy that it takes to do the second reaction. So you take these two things combined, and really the net for this entire two-step reaction is still exergonic. All you did was take minus 7.3 plus 4.0 kcal per mole. This gives you still an exergonic reaction. So the cell's like, all right, well, we have a little bit less energy in our cell. That's okay. I really needed this glucose 6-phosphate, so thanks. Okay. And that's not the end. The cell's like, now can you change that glucose 6-phosphate to something else, please? And then another enzyme's like, yes, I'll do that for you. Okay, and that's it. So just like I asked you to do with the proteins in general, these are common types of enzymes. So you have your proteins, that's your produce. Then you have your enzymes, that's your apples. Then you have other types of proteins like, you know, structural proteins. These are different types of pairs. You have immune proteins. These are your different types of oranges. So in the enzymes, you have all of these common types. Your Granny Smith, your Envy, your Jazz, etc. So your job is to be familiar with them. And so what that means is if I explain to you what an enzyme does, how it modifies a substrate, you should be able to identify what kind of enzyme it is. So if I said, oh, this enzyme uses water to hydrolyze a molecule of GTP, you would say, oh, that enzyme must have been a GTPase because it hydrolyzes GTP. Okay. So there you have it. All right. That's it, right? Yeah, cool. Questions? Okay, so Madeline, I think you maybe didn't get a question answered earlier, so I'm gonna go ahead and start with you if you still have that question.
Yeah, I just wanted to make sure I understood two things. One is just making sure the lock and key model, so that's just a rarer occurrence, correct?
Yeah, it's just not as common. And again, it's for that very reason that because it does happen, but because the substrate would have such a high affinity for the enzyme, it would take a lot of energy to release the products. And so that's not the goal of an enzyme, right? The goal is to quickly do this, release the product.
All right. And the second thing was, when you say affinity, does that just mean like how well it fits? Like
affinity is a chemistry term, and it's just how well the molecular properties are attracted to one another.
Okay.
So I guess like if you wanted to explain it to yourself now, you can say like how well it fits. But if I said, what does it mean how well it fits? And really what's happening is one surface of the substrate is interacting with the surface of the active site. How are those surfaces interacting? Via some kind of molecular interaction.
All right, that makes sense. Thank you.
You're welcome. Malia.
Yeah, I was just wanted to clarify, do we need to like memorize the different types of the enzymes with the like basic functions of them, or like on the test, like you were saying, you would ask like, oh, the GTPase, like you would just name it. So do we need to know like the exact name for that definition?
I think that the answer to this question is probably yes, because that by understanding, so let's go back to this, by actually not just memorizing, but understanding what breaking down proteins by hydrolyzing bonds between the amino acids means, by understanding what that means. So let's use this. So if I gave you, oh, there is a dipeptide and a dipeptide of, I don't know, arginine and lysine, okay? And this dipeptide was cleaved by an enzyme into its two individual monomers. What type of enzyme do you predict this molecule is. And you would know, well, I know, let's see, well, I know that proteases break the bonds in between amino acids. It has to be a protease. But you wouldn't be able to know that if you didn't memorize what protease is or understand what a protease is.
Okay, perfect. Thank you.
So yeah, so that just kind of gives you insight. I'm never going to say define protease and then you're just going to memory vomit this. I'm going to give you an example and then you're going to apply your knowledge to it to get the right answer.
Thank you.
You're welcome. Any other questions? Okay, awesome. Well, guess what? We're back on track. So I'll see the Thursday lab in just a couple minutes. And for everyone else, have a dry, cozy weekend. And I'll see you soon.
Thank you. Have a good weekend.
Thank you.
Thank you.